So without further ado, I'm going to show you some stuff. Okay. And as always, if you have questions, please let me know, and I will stay afterwards after office hours to answer questions as well. Okay, right. All right. All right, so these slides are what I want to talk about. This is really about, sort of about the material covered in chapter 12 of the textbook. But I'm going to talk about some papers and more recent work as well to try to give you an idea of what is there beyond GMAC-based protein design, what is there that can model binding, and how do these actually work? And I'll try to talk about both the computational and the kind of biological or biophysical aspects of it. All right. So what we probably understand pretty well now is the Mayo type papers, right? We looked at chapter 11, we kind of went through that in some detail. And, we went through this paper and we thought about it some, but what about more recently? So again, what are the fundamental concepts? Rotamers, obviously, we've been talking about those a lot. Now, in general, in any of these things, some of you ask what the workflow looks like a little bit. So generally, the workflow is going to look something like this, is that all these things are an input model here, right? This is not part of the algorithm. I think the computer scientist who came into the field, that's a major contribution, not to think that these are in the algorithms. The energy function, think of this as passing a pointer to a function. You have a structure here. Rotamer library is like a database. These all go into the algorithm. Then you make these predictions about specificity, stability, and so forth, right? That's kind of what we're doing here. And what are the big ideas in chapter 12 and as we think about protein design in a more modern sense? So the first is, as we've discussed, predictions made using rigid rotamers are very inaccurate. Proteins are actually flexible, but the main thing is that you'll get all this false precision where the rotamers are fixed at discrete finite values, when in fact they can move sort of continuously to adapt, find better conformations, and avoid steric clashes. So the real way to think about this is as follows. So biophysicists minimize things all the time. Like you're going to minimize slightly your structure, your PDB structure, before you do design on it to get rid of small imperfections. That's a modeling step. You're going to do your design. And at the end, before doing any analysis, you're going to minimize the predicted structure a little bit to try to get rid of these small changes. So you might minimize, let the rotamers, even the backbones, move a small amount. So here's the key thing. If you start out with some structure and you do your dead end elimination pruning or even Monte Carlo pruning, any kind of pruning, and you get a bunch of surviving rotamers and amino acids, and then you pick one somehow, maybe the best one, maybe something else, and you declare it's the best one. Now, suppose you take that structure and you minimize it again. So now you have a minimized structure, even lower energy. Now pick one of the solutions that you pruned. If you take that and minimize it with the same parameters, and you might be putting new rotamers, new amino acids on it, whatever. If you do this, it might minimize to a lower energy than your putative GMAC. So in other words, the prediction that you have the lowest energy structure does not take into account the fact that you're going to minimize it at the end. So this is a kind of deep thing. The dead end elimination as presented is not minimization aware. So this is actually a deep point in computer science. You might know that in GPU architectures, we have a notion of speculative execution where you predict a process that's going to run later, such as maybe a branch or a subroutine, and you are going to proceed down that path even though you don't have all of the data. So instead, we want to keep the minimization, which is good, but we don't want to prune things that would minimize to a lower energy than what we would get if we only had rigid things. So the idea is you have this enormous set or space of conformations shown here. I hope you can see my pointer move around some. And with minimized dead end elimination, you're going to prune this down to a small set. Like you're going to get rid of things that can't participate in the minimized GMAC. And the thing is, this should be provable even with energy minimization at the end. So in other words, we're going to prune rotamers, prune any rotamer that cannot participate in the GMAC, even given that after we compute the GMAC, we're going to minimize it slightly. That's the key thing. That's what minimization aware means. And second, we're going to consider ensembles. So in other words, we're going to take this set of conformations and we're going to compute partition functions over molecular ensembles in order to be able to estimate binding and stability and things like that, specificity. Now, this is a difficult computation, so you're going to see a new kind of algorithm for some of you all today. It's called an Epsilon-Approximation Algorithm. And so the notion is that while this process, before this process runs, as part of the input, and the input model is here, in addition to all this stuff up top, the user is going to give the program a value called epsilon. And epsilon is how close do you insist the answer be. So this is not a statistical thing. This is not an average. It's not some post hoc thing. It's something that must be enforced. So in other words, if the true partition function is Q, you want to be close to it. How close? Multiplicatively close. Your answer should be 1 minus epsilon times Q within a range like that. So lots of ideas here, right? Minimization awareness, ensembles, and epsilon approximation algorithms, which they're a mainstay of computer science, but they're usually not taught in our early discrete math and algorithms classes, but they're essential. And I'm going to show you how this is actually possible. All right, so let's look at a motivating example. This happens to be from my work, but there's lots of work like this. So this is gramocydin S. S is for Soviet, discovered by Soviet scientists around 1920 and actually used in World War I. It's a topical antibiotic. So what is this guy? So this is a non-ribosomal peptide. So it's made by enzymes called non-ribosomal peptide synthetases, or NRPS. These are found in various small organisms, and they make them as chemical warfare against other creatures that want to eat them or compete with them. And this includes, for example, vancomycin, penicillin, bacitracin, and so forth. And they're basically modified peptides, and these are made outside of the standard ribosomal pathway. So no sort of DNA to messenger RNA to proteins. These are like chemical factories. And if you look at this, it looks a bit like a protein in some bizarre ways. So we have phenylalanine, although it's reversed. This is the wrong chirality, or the opposite chirality, D-phenylalanine instead of L, which occurs in natural proteins. We have a proline here, we have a valine, we have an ornithine. This is a weird non-natural amino acid. It's not proteinogenic. There are about 200 of those. So these get incorporated, made in this modules, and then cyclized at the end. You notice it's basically a pentamer that's doubled here. It's a good antibiotic. It's highly toxic. You can't take it orally. So how do these enzymes work? Well, they work like this. You take in an amino acid, right? And you take in ATP and you adenylate it, which means you basically cleave off the inorganic pyrophosphate and you covalently attach the resulting AMP, adenosine monophosphate, to the backbone of the protein. And then these get incorporated into this chain here. And there are all these modules that are like little pieces of a factory. So adenylation is the attaching of the AMP to the backbone to activate it. Thiolation then attaches a phosphopantothene arm to this so that it then can be grabbed by the next module. Epimerization, this is amazing. This is a module. These are all proteins that flips the chirality from L to D. And the condensation domain makes the peptide bonds, a little bit like the sites in the ribosome, makes the peptide bond between the phenylalanine and the proline like this. And then things continue all the way down here. And then the thioesterase domain cyclizes it at the end. Again, adenylation, this is important to memorize. Adenylation is the reaction substrate plus ATP goes to 8 amino acid substrate covalently attached to AMP, releasing inorganic pyrophosphate. I mention this because if you go on in genomics, there's a form of adenylation, and many genomics people don't know the biochemistry behind it, but it's very simple. Again, the recognition or specificity is based on the active site binding the particular amino acid. So the kind of cool thing here is, well, could we modify this pipeline to make new antibiotics, right? That would be pretty neat. Have a combinatorial factory to make these using the same natural enzymes that make these in fungi, bacteria, and other small protists. So the idea would be something like this. Here's our natural modules. There are two modules here, GRS-A, GRS-B, for gramocydin synthetase. Here's a phenylalanine adenylation domain. What if we change that to adenylate a different substrate? So we change that to adenylate leucine. Wouldn't it be nice if it gets passed down all the other modules, then we get leucine incorporated in our chain? or alanine. So we can make all these different things, and that requires redesigning the enzyme, which is a natural enzyme, to adenylate a different product, say leucine, instead of phenylalanine. Again, adenylation being this reaction. So we're redesigning nature's machinery here to our own needs, kind of a synthetic biology aim. And you could even replace, here's another example, you could replace the, down here, you could replace the ornithine with the glutamic acid instead. So we could diversify this by enzyme design, which is a very promising thing. So I'll work through a little example here, maybe later if we have time. The combinatorics of this are a little bit large. So here we have crystal structures of this phenylalanine adenylation domain. And so you see all these residues that kind of touch. This is a cartoon of those structures. You can see it. Here's the phenylalanine substrate. So this is where this is a protein that takes an amino acid as an input and produces a peptide, although not a standard peptide. So this is the input here. ATP would be up here. And basically there's this asp and this lysine here, and they basically coordinate with the backbone of the amino acid. And all these other residues here confer specificity and binding. So the idea is, well, what if we want to swap out the phenylalanine here, and instead we want to put in the leucine here like this? If we do that, we might consider changing any two of these at one time. There are 9 different things in the active site. So the combinatorics is given to us by an active site of size r equals 9, changing s equals 2 things at once. And if you look at this and take out some things that are obviously not sequences you want to try, we might just look at mutations to these 9 residues that are compatible with a small hydrophobic amino acid like leucine, such as glycine, alanine, valine, leucine, et cetera here. This gives us approximately 3,000 mutation sequences, about nearly 750 million conformations. Incidentally, iMinDEE prunes this down to merely 80,000. So, presumably 3,000 is more things than you want to make in the lab. When I have enzymologists working in my lab, they want to have good enough predictions to try 10 things for their thesis. They don't want to try 3,000. So this is the combinatorial problem we have. And notice it's discrete. We're sticking in different amino acids here that are going to change the specificity. So when I swap out phenylalanine to leucine, I get activity and binding. All right, that's the problem. So that's pretty cool, but there are all these questions here that we want to answer. And one is, how do you model flexibility, right? These are flexible things. So to give you an idea about this, and this is really about continuous flexibility in some sense. Let's look at what a peptide actually does. Here's a molecular dynamics simulation of a peptide in solution. So these things move around quite a bit. And so that, any model where you treat things as rigid probably needs to be enhanced to get good predictions. And this is a primary biophysical justification for thinking about things like continuous flexibility and having things be more flexible. So it's kind of obvious we have to think about how to model that, but that will be a modeling choice. We have to think of what is the best way to do this. All right, let's go to our slides again. All right. Is it amyloid peptide? All right. So again, we're going to go back to our picture here, and we're going to say that, we'd have rotamers. We're going to change the rotamers, but we're going to let things be flexible here. So the rotamers can be changed different amino acids. They can be flexible here. Again, the rotamers will be either the low energy wells of this configuration space, or there'll be statistically significant rotamers where you could view these peaks and valleys as probabilities, and we could subsample them. So this is a discrete approximation to continuous flexibility here. We could subsample the rotameric space for a tyrosine like this and get something finer that's still discrete. Okay. And again, I want to contrast how we model flexibility with this slide I had before. So on the left, we have the computer science way of thinking. The maximum likelihood formulation in computer science corresponds to the GMAC in biophysics, namely the global minimum energy configuration or conformation, the lowest energy, the single one conformation that has the lowest energy. And if, among your states, that is the most probable one, because we move back and forth between energy and probability using the Boltzmann distribution, e to the minus EC over RT, some of those things. But of course, we have many conformations. These are rapidly interconverting in vitro or in vivo. And so this corresponds, roughly speaking, to a quote unquote Bayesian approach, average over conformations instead. And this is some kind of integral or sum perhaps this integral over all conformations is what we call the partition function. And this boils down to binding for this. We basically want to calculate the probability in the model of being bound divided by the probability of being unbound. And if these individual conformations are predicted ensembles are our microstates, this corresponds to a ratio of partition functions. The probability of being bound, this is a set of bound structures, is this sum. Here's the free protein, here's the free ligand, and you divide this, and this is an approximation to by Ka, which is 1 over Kd, the binding constant at equilibrium. So we have this problem of computing this and estimating it for all of our different mutations that we want to think about. And there are a lot of mutations, and this is a large computation because this is a very big sum. Okay. Now, moving along, what do we know? So for protein design in this NRPS pathway, we have structures, which is great. And we can visualize the active site here. Here's phenylalanine in a space-filling model. And we can develop cartoons and mnemonics like this, like I showed you before. So our goal is to change, say, any two of these, up to two of these at once, consider lots of different mutations, and therefore mutants, and to predict the binding of the substrate, which is leucine, instead of phenylalanine, and see if it will work. So what's kind of cool about this is we basically reduced this problem to, which seems really hard, like how do you redesign enzymes that make antibiotics? We've reduced the problem to a large scientific computation problem. Is how do we compute these ratios of partition functions? How do we do that over all possible mutants? And in particular, we don't want to just enumerate all these mutants. We want something more like DEE. We want something that you can prune things so you don't have to look at the entire search space. So the magic of DEE doesn't have to look at 1 trillion different possibilities. Similarly, an algorithm that computes K-star shouldn't have to look at 1 billion different mutants, let alone conformations, in order to get an approximation. So that's the algorithmic challenge. So at this point, it's in the hands of computer scientists who understand a little bit of biophysics. How can you develop such an algorithm? But let's look at some things. What do these ensembles look like? Well, I could draw you pictures where things are overlapped like this, but here's an interesting way to think about it. So here, I've just taken the wild type phenylalanine adenylation domain, or Phe, and I've just created an ensemble, minimized ensemble with K-star. And here we've drawn the, each point in the ensemble comprises an entire structure of the active site, and I've plotted it here. It's predicted energy, and it's RMSD to the wild type structure. And so what you can see here is that a couple of things. One is, if you truly wanted to get the GMAC, like what do you pick? Like the GMAC is this one here, but are you really sure it's better than this one? And you're really sure it's better than this one? And besides, this is sort of the right answer. So the GMAC is very brittle, right? I mean, it depends on these energies. And these differ significantly. There's different rotameric choices here. So if you're forced to pick the GMAC, you have to be really precise. There's all this possibility for false precision and brittleness. Whereas in the ensemble model, you're essentially saying, well, any of these are pretty reasonable. Down here, maybe that's my cluster, and I'll just take a partition function over those things. Now, there's some other things to notice. This figure is, from a pretty old paper of mine, and my wife is a neurobiologist, very quantitative. And when I showed her this figure, she said, "You've got a bug in your program. Like, look how these patterns repeat. Look how these kind of look similar, but just shifted up in these different ways." Clearly, there's an artifact, because biologically, there's no way you'd see a pattern like that. I mean, that just wouldn't happen. And I was like, oh my goodness, I didn't think of that. I just thought it was data. But then I realized what the true story was, and it's not as bad as that. What do you think the issue is here? Is there a problem, or is it okay? Or if I just tell you that it's okay, why would this be okay? Why would we see a pattern like this? Any ideas?
Could you explain again what the clusters represent? Or I guess that would be giving away the answer, maybe.
Oh, no, well, I'm happy to do that. I would like to tell you rather the what a point represents. right? So a point represents a structure, an entire structure predicted from K-star algorithm, right? So it's a complete structure with 3D coordinates, all the rotamers assigned and everything. Just one sequence that we're looking at conformations here. And so for each structure, I can compute its energy, right, in the theoretical model, energy function, and I can compute its RMSD to the wild type, right? I start with one rigid structure, right? So each point is the structure. And I can't tell you what the clusters represent, right? That's sort of the data analysis problem. But the question is, why would you see these sort of repeated patterns? And is there a problem with that? Does that help?
Yeah. I guess I can take a guess. I think maybe if you change like one or two of the rotamers or two of the like I guess variables, but everything else stays the same, it would make sense that it was just like sort of shifted over a little bit.
Yeah, that's the right answer. So we call this rotamer ratcheting. So in other words, if you imagine, I'll wave my hands here. If you imagine you have an active site here, and for this rotamer, I could have, say, a leucine that could be like here, can I do here, here, or here, 3 positions. And over here, I'm changing something else. So I have, I choose one rotamer here, and this one goes through 3 different positions. Then I put this one up to 2, and this one goes through the same 3 positions. So the combinatorial sequence repeats basically, and I just shift them up and have different gaps in energies depending on what goes on. So the rotamer ratcheting actually is a kind of a, I mean, it's not unreasonable. It's kind of an artifact of the discrete, rotamer model in some sense, although these discrete rotamers are minimized. So this is actually okay. But so great answer. I think you got it. But the main point here is that, from our point of view, when we think about this, I guess what I would say thinking about this problem is that I guess the way I would describe this is it shows the fragility of the kind of GMEC way of doing this, right? Because you don't really know what to pick down in this lower left-hand corner. Like you don't really have a good model for that. So we really have we really don't understand which one. There's no criterion for picking these. It's sort of an fairly arbitrary choice. And so one of the things that the ensemble model really gives you here is a technique whereby you are going to be able to combine the information in a reasonable way without having false precision, which I think is, a pretty cool thing. Does that make sense? Let me know if you have any questions on this. Is that pretty clear for everybody? Okay, well, let's go on. All right, so how can we do this without looking at everything? So similar to DE, we need some technique for pruning things. So one thing you might think of is sort of steric pruning. Like you can basically look at things that, have a steric clash even when unminimized. The key thing about this kind of pruning is that, and this is, this is a tree. So what does this tree mean here? So these different positions, A, B, and C, are different positions in the protein that we're designing. And the different branches here, like 1, 2, 3, 4, these are different rotamers you might consider putting at position A, and these are different rotamers you might consider putting at position B. So the main point of this figure is that when you prune something, say here, this number 3 or this number 2, you prune all things that would be in the subtree below it that would contain that rotamer. And so as a result, you get a considerable speed up, maybe not when it's really far down here, but when it's way up at the top, because this tree can be polynomial or even exponential size in relative to the size of the design problem. So when you prune something up here, you're not only pruning one thing, you're pruning all possible proteins in some sense, all possible sequences that would have that rotamer and all their different combinations. And so for that reason, this is a significant source of speed up. So that's sort of the intuition between how we can get some pruning. All right, so I want to now describe why, given this big problem, it's even possible to get approximations. Like, why would this even work? How can we think about it? But before I do this, I want to kind of reassure you and give you a little math here. A little math path. Math path is that I like to call it, a path to our math. So why are we climbing this? So it's a hill, but I'll help you up it. So we always want algorithms with provable accuracy. Otherwise, we don't know what we're computing, right? Now, some examples that we talk about. The original K-star is here, and we can read more. There's versions of this that are faster or more accurate. There are versions that incorporate backbone flexibility in a very rational way. And there are techniques that we have at least one project based on this that is using sparse interaction graphs rather than complete interaction graphs. And this gives you the possibility to use dynamic programming, which means that in certain cases, your problem isn't going to be worst case exponential, but rather worst case exponential only in sort of a branching factor in the graph. So you want examples of these things that have these guarantees. So we always want accuracy, right? That's great. But what about how fast these things are? And the reason, computer science is actually a science is that the complexity bounds are predicting what would happen if you ran the program even before you do it. So it's just like science, right? It's making a prediction about what would happen if you ran the program ahead of doing it. And you can, it's science because you can then measure it and see if that was correct. So here are different kinds of improvements we could have. So we can have a constant factor speed up. So if the basic program is going to take f of n, some function of n amount of time, we could reduce the constant. And this could be very significant in practice. Generally, theorists aren't as excited about it, but making c much smaller than d, could be remarkable. It can make the difference between being able to do a design and not. And that's what I'm talking about today, mostly. Now, we also might want a combinatorial speed up. This is even better. So this is like the case where we pruned an entire subtree of our search tree. So depending on how big the subtree you're pruning is, think of game tree, right? I mean, you've got your queen out in chess, and one possible crazy thing you could do is just to sacrifice your queen. Now, if you're a genius, that's probably part of some important gambit, but for most of us, especially at my level of play, that's a suicidal move. But there's an exponential number of possible moves up to losing after you sacrifice your queen, and you're pruning all of those boards, right? So you could get an exponential reduction in the size of your search space by pruning things that are obviously bad, such as sacrificing your queen or something with an irresolvable steric clash. Or you still could get a big speed up. I mean, even if you have a big search space, being able to reduce your search space by a polynomial amount, that still is quite remarkable if you could do it. It's better than having to look at everything. And of course, you could imagine, so this is our example, again, if you prune something way up here because of a steric clash or some bad interaction, you prune the entire subtree down below this, which obviously is exponential in the depth of the subtree, namely the number of residue positions. Remember, A, B, and C are positions on the protein. 1, 2, 3, 4 are what you're changing it to. Okay. So coming back to this combinatorial speed up could be something you empirically observe. You could show that it's possible, and then how much speed up you get depends on the problem. An even stronger version is one to say that you always get a provable speed up, like you always can speed it up by this factor, and that would be quite remarkable. So these are different things we might try to prove about complexity. So again, I mean, why would we even consider something without provable accuracy? I mean, otherwise you don't know what you're getting. That's the whole point of computer science. But we can say a lot about complexity as well. And what we can do here is we can get various kinds of complexity, various strengths of speed ups, and try to prove them so we know ahead of time how fast the algorithm will run. All right, and some examples here. Today I'll do constant factor speed up, and I'll hint at why you can get a combinatorial speed up. But I will go through this in some detail. Okay. And hold on just 1 second. There's some interference on my line. I'm gonna try to deal with that. Okay, we're good now. All right. So we won't do all of this today, but I'll give you a little hint of it.
Could you could you explain the notation after the combinatorial speed up again?
Yeah. Tell me what your background is. I'll tailor my answer to you.
Biomedical engineering.
Okay. So does this notation here make sense? Order 2 to the n?
Yes. Or
Okay.
so does that mean you could speed it up as a 2 to the n polynomial?
Sorry, you asked me at the left or the right?
I'm like both of them. Sorry.
I want to take them separately. So I'm trying to break down your question into 2 parts. Let's look at the right half.
Okay.
Tell me if you understand this notation.
I do not.
You do not? Sorry, you dropped out. Can you say it again?
I do not. No, I don't. Sorry.
All right. So this is, I'll offer several things. So this means that it's the size of this is essentially bounded by a function that grows as fast as 2 to the n. And this means it's bounded by a function that grows as fast as some polynomial, but you don't know which polynomial. Does that help?
Yes. Thank you.
Good. And let me offer some, I'm happy to talk with you about this offline. It's a very useful thing in engineering as well as computer science. But if you're interested, I think I made available lectures on this from our first year course in complexity. And I believe this is lecture 05 that's on the web. And feel free to take a look at that. There also are some notes. And if that's not clear, come talk to me and I'll help you out.
Okay, sounds good. Thank you.
So in words, this would mean a polynomial speed up. This would mean an exponential speed up. Reducing the size of the problem by a polynomial, although you don't know which one, or an exponential. Great question. Feel free to keep asking questions. All right. So let's now get into some details and think about why should we get an approximation? Remember, we want to compute this huge sum of exponentials. So let's write it out like this. So there is a good one, there is a smallest one, right? Let's say we get the smallest one here, maybe it's just a decent one here. So we have a smallest energy, and then we have a bunch of other energies. We exponentiate these and we add them up. I'm leaving out the normalization factor by RT. So the point of this is, what's the intuition? Why can we get an approximation? So I'm going to rewrite this just to pull out the smallest one like this. The smallest one's now out here. And now I have 1 here and a bunch of other things. How big are these other things? Well, the thing in the parentheses are all non-positive, less than or equal to 0. So therefore, the exponential here is less than or equal to 1. So you have 1, and then a bunch of things less than or equal to 1 here. So we've just rewritten this. So all of a sudden, computer scientists get excited because we have our first bound, although it's very loose. We can bound Q roughly by the number of confirmations times the exponential of the smallest one, because these are all less than 1. So we have a rough bound. How could we ever use such a thing is another question, but this is our start. So that's our first idea. So look, we can bound things. So the point is, when you have approximations, you don't count things exactly, you bound them, and then you add up the bounds and you prove that the sum of the bounds is within something reasonable, so you can still decide an answer. Okay. Now, our situation is a little different. This is just math that describes, various sums. Suppose we're actually running our big sum here, right? So we want to compute Q, which is the true partition function, the true sum of these exponentials. Suppose I've let this happen for a while and you spent a couple of hours and you've computed a partial sum Q star. This is like what we call a running total. So in other words, you've summed up a bunch of stuff, you've written it on a napkin, there's a bunch of other things at the door waiting to come in, you haven't summed those yet. So Q star obviously is your approximation to Q, right? There, so that's fine. So we have this thing here. So I know I have n things that I haven't looked at yet. Well, I can bound these I can bound these roughly. So suppose all of the things I haven't looked at have an energy above E sub t. I don't know E sub t yet, but I give it a variable name. That's what algebra is all about. So I can bound Q above by Q star my running sum plus n the number of confirmations left to see times x of minus E of t, where ET is the energy is at least E of t for these guys. So that's what I have, right? Remember, I don't know Q. I know Q star because I've computed it and leaving ET as a variable. Now, here's the thing that we do frequently in computational work is we write down what we would want. And since we're mathematicians, we can write down anything we want as long as it's consistent. So what I want is this. I want to know that my running sum is very close to the true sum. So I just stipulate, I have the user give me an epsilon, and they can't change it. They have to give me an epsilon small probably. And I'm going to say, okay, I'll respect your epsilon. What I want is Q star to be greater than 1 minus epsilon times Q. So in equation 1, I have what I know. In equation 2, I have what I want. So I just combine them and I substitute in. So substituting this into that for Q star, I get the following equation. So basically, now I have my running sum is greater than equal 1 plus epsilon times this quantity here like this. So I got multiple Q stars. Obviously, you have to simplify. So now I just do some algebra. It's all just standard algebra. I rearrange things a little bit like this. So I got my epsilons and Q stars here. I've got everything here. So what's kind of cool about this is that I know most of these things. Let me do a little more simplification. I'll put a ratio called rho here like this, substitute and take logs. And now I get this equation here. So look at this. I know Q star because it's my running sum. I know rho because you gave me epsilon. I know n, the number of confirmations I haven't looked at yet. I don't know E sub t. So the whole point about this scheme is I want to not look at things that have a certain energy, E sub t, but I know what E sub t is. So now I get a pruning condition. I get a condition that says, hey, if you know that any confirmation in the future has energy worse than E sub t, you can simply ignore it. It won't affect your epsilon approximation. Just forget about it. So we can ignore any terms of energy satisfying this equation. So this is pretty exciting from the algorithm point of view. We have an actual pruning condition almost. All right. It's very simple derivation, just requires a little thought. All right. Now, the problem with this is, how could we know a confirmation has energy worse than E sub t without computing the energy, right? That seems circular. So we need a way around that. And again, bounds will be there and the time to compute the bounds. All right. So computing the minimized energy. Remember, minimized energy requires minimizing these things. That's the whole continuous Rotamer thing. And actually, then that takes time quadratic in the number of atoms, like m is the number of atoms, times the number of iterative steps. You have to iterate this thing down. We don't know what I is really. So this is a very expensive operation. Your protein could have thousands of atoms in it, right? So actually computing E sub t is expensive. So what we want to have is a technique that very quickly computes a lower bound on the energies, right? And time quadratic merely in R, rather than quadratic in m squared times this number of steps, which we don't even know. Remember, R is tiny, it's the size of the active site, m is enormous, it's the number of atoms. And if we can do this, our claim is that we'll actually have an epsilon approximation algorithm, namely that our running sum bounds from above the actual partition function, and then that'll be close enough. So I need to tell you a quick way to compute a bound on the energy that is cheap, that's in yellow, and I can use that instead of computing the full energy. So basically, if the bound is satisfied, I just don't have to do very much. If it is not satisfied, I have to compute the energy. So only compute the energy for things that are close by. Again, remember, while we're doing this, rotamers are actually continuous. The energies are very sensitive to small changes in conformation. And in this model, if you want to kind of think of this kind of categorically, our model in rigid rotamers is that in our chi1, chi2 space, which is mapping dihedral angles, chi1 versus chi2, the rotamers are points. They're very brittle. Our model now is that rotamers are now points in squares, right? So the model is the discrete part is you say which square, and you let the algorithm decide the continuous position within that square. And that relieves these tiny clashes, which would give you enormous energies. This conformation here in the rigid rotamer model would get pruned by the pruning step and never considered, even though once you minimize it, you might have beautiful contexts like that with excellent energies. That's the whole idea, reason we're doing that, right? All right, so how do we do something like this computationally? Here's a little cartoon of an active site with various rotamers here. So the key thing about this is that these rotamers aren't rigid. They can flex a little bit. Each dihedral angle here, as you can see from my mouse, flexes plus or minus 9 degrees, plus or minus 9 degrees in all these angles. And what you'd like between this pair is the minimum configuration in that voxel for this pair of rotamers. This one is minimized, this was minimized. So you're computing the pairwise energy here, Eij. This is the minimum pairwise energy. So because we have a pairwise energy function, and this assumption is crucial for this result, the total energy is always greater than the sum of all the minimum pairwise interaction energies. So what does that mean? The pairwise interaction energy that's minimized means, look at the energy between these two here, the two-body energy. Now minimize that over the mutual voxel for I and for j. In other words, back in this picture here, we're allowing one rotamer, you're picking a rotamer, but we're also allowing minimization within his for one of the rotamers, and for the partner rotamer, the same thing. Wouldn't be drawn in the same graph, but that's the same idea. So we're picking the lowest energy state here that accommodates the other one. So we look at the, so again, this bounding is super important, and I've actually written the language uses this. The total energy is provably bounded below by the sum of the minimum pairwise interaction energies. That's the kind of key little theorem here. It's the theorem about pairwise energies. So what do? Well, we compute them all. You could pre-compute all the minimum pairwise energies and you put them in a table. So this isn't so bad, right? So basically, if you have a bunch of rotamers like this, right? This is basically essentially quadratic time to do this. So the pre-processing of this is only going to be quadratic time to compute this guy. So that's pretty neat that you can do this. So basically for every pair of rotamers, like the red one, the orange one, and so forth, what you're going to do is you're going to minimize that pair together in the same voxel with nothing else in the active site. So all these will be converted to alanines, so don't get in the way. Get the lowest energy for these guys, stick that energy in a table, and we look these up in a table. So now we have a pre-computed table of bounds on pairwise minimum energies. And now we're done. Basically, in order to compute the bound here that we needed in this case, to compute this bound, all we need to do is sum these up because everything is pairwise. We just look up the appropriate energies here and sum them up, and we can do that rather quickly. That's pretty nice. Okay. So what does this mean in terms of a criterion like dead-end elimination? I won't derive it here, but it is in the paper. We had all these terms in dead-end elimination. We actually went through them in, I believe, my second presentation. And now we have a more complicated equation. So instead of having rigid energies, we have bounds on energies. So we have something similar to what we had before, lower bounds on basically conformational energies. That's similar to what we had before. Then we have upper bounds on other kinds of energies. So these terms are quite similar to what we had before. We have a new term here, which is possible energy changes due to the rotamer movement. And you can work this out. It's not in traditional DE. We have to add the term. But then we can make the jump from rigid to flexible rotamers, which adds a good amount of flexibility to our designs and makes them more robust. So same kind of argument, but just more complex. And finally, this can actually be extended in certain ways. So how does this actually work now that you know a little bit about A-star, or you will by Wednesday? So again, we have a new criterion that will prune on bounds. So remember, A-star search with rigid rotamers will enumerate solutions in order of energies gap-free, starting with the lowest energy. Remarkable thing. And if that had been available to Steve Mayo and his colleagues, instead of using Monte Carlo, they might have used A-star search, and then that table of 1,000 conformations, instead of being generated by Monte Carlo's processes, could have been generated by an A-star search with provable enumeration. Here we'll do that, we'll use that algorithm, except it'll be a little different. So we don't have access to the true energies, we only have access to lower bounds on the energies. So what A-star search will do in this case is it will enumerate rotameric solutions in order of lower bounds. So the first thing off the tree will be the thing that has the lowest energy bounds. Remember, bounds aren't the true energies, they're just lower bounds in the energies. And then the second one will be the next one, right? The next lowest bounds. The third one will be something that has the third lowest bounds. But here's the thing. As you take these off the tree, what you do now is a full minimization. So you're just doing this full energy minimization that takes quadratic time times time steps on the best candidates based on their lower bounds. So the full energy minimization gives me a beautiful minimized structure, and then it also is going to give me a lower energy because I've done the minimization. And now I put this in my data structure here based on the minimized energy. So in this simulation cartoon, the first one comes off with the lowest bounds. I minimize him, he goes in the tree. Second one comes off, and just for this example, and minimize him, he also goes in the tree. The third one's going to be in white. He's going to have the third lowest bounds like this, but when you actually minimize him, he minimizes even lower than the other ones, which is pretty cool. So he goes up to the top and so forth. So in this process, we fill this out and we stop when the bounds of what we pull off the tree are provably worse than the energy of our best guy. And then we have the GMAC plus a lot of other confirmations that we got. So we have to minimize until we get someone whose lower bounds are greater than the energy of the best thing in my queue. And this is the minimized GMAC, right? So we have a technique now that's minimization aware to compute at least the GMAC and gives us some other things as well. That's kind of a neat thing. Now, these techniques give you GMACs, either the minimized GMAC or the traditional GMAC, kind of like a maximal likelihood solution. But we'd like to do this business where we compute binding, right? That would be really wonderful if we could do that. So how would we do such a thing? Again, we want to compute something like the K-star score from these based on a weighted average of these confirmations. That's sort of our key idea here. So this is the height of complexity for this lecture, but I want to give you an idea, especially because many of you asked about this. So going back to our idea here, you see, but by the time you've emitted the GMAC, the minimized GMAC, you do have all these other low energy confirmations here, right? They might be pretty decent. And so that's sort of the idea that, well, why couldn't we use these to make a K-star approximation? We have low energy confirmations, and we know the partition function will be dominated by low energy confirmations with energy, say, EC, because it's their contribution statistical weight is e to the minus EC, so the lower, the better. So can we do something like not just take the best one, but take these guys here and put them into a K-star calculation? So how can we do that provably? Let me try to answer some questions we had. So here's the scheme. So first, yes, we'd filter some stuff out and we get a big sequence space like this. Now, as we had before, our DE or MinDEE type pruning is going to give us a subset of the rotamer confirmation space, right? So these are the things these are basically the set of rotameric choices and amino acid sequence choices, but let's say rotameric choices that have the possibility to contribute to the minimized GMAC. And the red things here that are pruned out, these are the things that are rejected, this huge number of things. These are things that cannot possibly contribute to the GMAC even after minimization. So in other words, the red things cannot participate in the GMAC confirmation or sequence. Pretty important thing there. For purposes of exposition, I'm just going to assume we have a single sequence here. This can be generalized to multiple sequences, but let's look at one sequence. So rephrasing this, C is now confirmations. It's a confirmation space. These are the confirmations or an implicit encoding thereof as factored product that can participate in the minimized GMAC. These are conformations or encodings of conformations as a factored product that cannot possibly contribute in the GMAC. That's done by the iMinDEE criterion I gave you before. Now, A-star search proceeds and it enumerates conformations out of this representation in order of energy lower bounds. And as they come off the queue, what we do is we do full energy minimization of those guys, and we add them to the partition function. How? Well, if you get the first one, the queue is initialized to 0 plus this exp here. The second one would be that plus the next one and so forth. So we just sum them up in here. Now we have an approximation to the partition function. Now, as mentioned in the comments and also in the paper, it is possible that the following could occur. Two possibilities. One is once you've enumerated all of these, you can tell that you have an epsilon approximation algorithm. In this case, you're happy. The algorithm outputs the answer. It opens a bottle of champagne and shuts down for the night. But it's also possible that you might not have an epsilon approximation algorithm. And that's because you prune so much that there are not enough conformations in here to have adequate statistical weight to approximate the Ka score. Why might that happen? Well, to say it baldly, the criterion here is whether or not a conformation can possibly participate in the GMAC. But the partition function doesn't really care very much about the GMAC. The GMAC is like the animals in Animal Farm by George Orwell. All the conformations are created equal, but some are more equal than others. So it's the best conformation, but so what? There are lots of other conformations. So the criterion of not being able to be in the GMAC might prune out some suboptimal conformations that would have significant statistical ability to contribute to the partition function. But in that case, because it's a provable algorithm, the algorithm knows this. It goes, oh my goodness, I've computed a nice partition function, but it's not epsilon close because I pruned too much, right? I pruned so many things that this isn't big enough. So I'll tell you a story now. Story time, as they say at TikTok. So my students came to me and they said, this is what we got. It's not epsilon approximate sometimes, and we can detect that. And I was like, oh, geez, yeah, I was hoping for a precise combinatorial algorithm. But I guess it won't be, but this is computational biology. So can we have a rigorous iterative algorithm? In other words, can you go back with the information you have and repeat the process, take some things out of the red prune region, stick them in here and run it again? And can repeat that, not too many times, repeat that until you get a provable approximation algorithm. So iterative algorithms are perfectly valid in computational biology, and in computer science, if you can bound their performance, no problem. So my students went away and they worked really hard, and they came back and they said, we think we can do this, but we want to ask you the following question. Is it okay if it only iterates twice? I was like, what do you mean? He says, well, what I mean is we can iterate, but we can prove that after we've iterated one more time, then we're guaranteed to get it. So this is an iterative algorithm, but it actually is a two-pass algorithm. You can prove it only needs to go back once. And the idea is something like this. So in this case here, as I mentioned, the nominal case where we basically go from enumerating just one thing to a whole bunch of things, we enumerate a lot of things here, and we get a good partition function, we're totally happy, right? And the reason we know we're happy is that although we haven't computed this partition function of the red stuff, we can bound it very efficiently. We can say, yeah, it's small. It's a loose bound. It's what we call a delta bound or an epsilon bound, but it's a provable bound. So in the best case, we're happy, but in the worst case, we don't have a partition. We don't have an approximation that's provable, right? That's the thing. So this best case is what we want, but sometimes that doesn't happen. So in the case where we don't have an provable approximation algorithm, we can take the information from this close but no cigar approximation and use it to run things once more. What we do instead is we take the value of P prime, and we use that to move some things from the red region into the green one. I make C bigger here, make this smaller. Therefore, this partition function will be larger, this one will be smaller, and then we can do that. We can prove how many we need to move from red to green, and after a second iteration, we're golden. And the algorithm actually is pretty straightforward here. So now what we do is we have a technique here where we repeat the search, but only once. In other words, it's a repetition in the code, but you can prove it only happens once. And then, of course, in this version, you have to run this for each sequence, and getting around that generalization will be something we'll talk about later. Okay, so that's the strategy there, and you have to prove this, of course, but that's the claim. Okay. Now, all of a sudden, we have generalized to minimized GMX. Pretty cool. Minimization aware, and we've been able to change the dead end elimination criterion so that it accounts for this continuous flexibility. And we can even generalize this in other ways. So we can put in backbone flexibility, have bounding boxes or back rubs, and we can design with that as well. So in other words, you could have flexible backbones as well for this. But I'll leave that to something you can read on your own or perhaps for a project. Okay, so in short, there's this choice of different algorithms, right? You can have rigid rotamers, continuous backbones, and you can compute these partition functions which actually model the binding at equilibrium in the model that you have here. Kind of a cool thing. So the way to think about this is that you're pipelining either MinDEE or one of these backbone algorithms through A star into K star to compute these ratios. The basic idea is that many conformations are pruned, but you save the ones with highest statistical weight. So you provably get close, i.e. epsilon close to the true partition function when you sum this up. And this means you can run this on thousands of sequences rather than just a few. And another feature of this kind of work is that what we can prove is less than what we observe in practice. Namely, we can pick epsilons such that in the model, we're guaranteed to get an epsilon approximation. But in fact, when we actually run this and compare it to some full partition functions, it actually converges much quicker. So in practice, although we don't usually use this because we want provable things, it actually is converging much faster than what we can prove. So a beautiful thing to work on in the future would be trying to strengthen the proofs to say that they converge with fewer conformations. So as you can see here, you can look at these scores here, and you can see that how many scores were needed to fully evaluate a partition function. It's a very small fraction of the conformations, far lower than needed by the bounds. So the bounds are a very rigorous way of defining how far the computation has to go. It's probably doing more work than it has to, but we can't prove that at this time. And finally, this lets us be a little bit close to our dream of making the protein design algorithm, something that's provable, so that differences between experiments and predictions are things that you can then go fix in the model because you know where they are. And I've already shown this slide, which is why we're going to get lots of good predictions here. Again, the difference between things like Monte Carlo algorithms and provable algorithms, that you get all the solutions, and there'll be lots of good things in here to test. That would be exciting for you to look at. And I want to show you a little bit of that empirically in the second half of the class. All right, so I think I'm going to skip how we generalize this to multiple sequences. I'll let you read about that. And given that it's 2:51, I would like to take a break and then show you some places where we can apply this experimentally in order that we can kind of see how it works now that we've done some algorithms. So let's take a 3-minute break, and I'll get some more tea, and we'll come back and we'll talk more about a little bit about algorithms, more about how you'd apply this in different cases and what you get out of it. Okay, great. So I'll see you in 2.5 minutes. All right, we're back. How's everyone doing? A lot of things to do today. Are you ready for me to go on?
I do have a quick question.
Absolutely.
Yeah, so when we're talking about like running it again or like with pruning less, is that by changing like the EW parameter that's kind of like the tolerance basically of pruning?
That's certainly one way to do it. It turns out that there's some more sophisticated ways that later evolved, but that's basically the concept. Yeah.
Okay.
So you just go further up, and you can set that algorithmically. In other words, you because you have bounds for P prime, which is the partition function of the prune things, you know how big that contribution would be. You can then estimate how many conformations you need to add back in. And that will let you set that tolerance, in fact.
Do they, when you're usually running it for the first time, do you set EW equal to 0, or do you usually have it to some value just kind of as your first time, making it more likely for that to succeed?
Well, you would never set it to 0 for a K-star calculation, right? I don't think, because you always want an ensemble.
Yeah.
Yeah. So basically, the notion of EW has been kind of superseded by having an interval parameter I in the algorithm called iMinDEE, which some of you discovered and discussed on Piazza, which was nice. So the idea is that what you're trying to estimate is the gap between the lower bound and the minimized energy. So the basic issue here is that the bounds are quick to compute, but they're extremely optimistic. So they generally think that a conformation can minimize to a much lower energy than it can. And the reason that's deleterious in terms of complexity is that then you have to enumerate more conformations until you defeat that bound and get something that's higher than it. So tightening those bounds is kind of the fundamental computational place for improvements in the future. But by being clever about that interval, which kind of replaces the notion of EW, it's possible to choose it in such a way that the performance is reasonable. But basically, EW is indeed your window, no pun intended, on how you would tune that. And the goal is to have a calculation of what EW should be based on the information about how far off your epsilon approximation is and how far off your how large the contribution of P prime, the partition function for the prune conformation, is. That having been said, it's not a theoretical concern in general, but it doesn't happen that frequently that you have to go back twice. But it probably happens in every design at least once over thousands of sequences. Make sense?
Yeah, cool.
Yeah, good. All right, so let's look at maybe some examples here. Let's see how this kind of works in practice. Show you some examples of using this. I think you get a little bit of feel for this. Go back to the NRPS example here. All right. So what can we do to kind of see that this is reasonable? One thing that people often do this here is what's called structure recovery, which is retrospective. So, I mean, we can use the same, our goal is to redesign this, but what we can do is we can say, well, what if we try to redesign it just to get the native structure back, right? So remember, when you're doing this, you're taking out all of these side chains, right? And then you're maybe changing a couple of different amino acids and you're asking to put all of the side chains back. And this is called, Osprey-based sidechain replacement. But the point is you actually are doing a structural prediction here. You're probably perturbing the backbones a little bit. You're replacing the configurations of all these side chains. So let's just do that for the wild type. And what do we get? And here's what you get. I mean, you get something that's pretty decent. I already showed you the statistics of this based on the ensemble, but based just on the GMAC of this, the predicted structure. So what you see here is you see predicted structure based on, in yellow, the X-ray structure is in blue. And of course, in this model, the ligand or substrate can actually translate and rotate in the active site as well. Here's AMP modeled in here. So most of them are pretty close with the exception of this tryptophan. That's actually not due to the rotamers, it's due to the fact there's a bond angle here that's different in some versions of tryptophan, and that gave us a reason to actually correct that in our rotamer library. So that the angle in this plane is off a little bit. But the rest of them are pretty close. So that gives some confidence that you're getting something pretty close. You can predict these active site structures. How about some perspective results, things that haven't been tested before? So how would this work? Again, remember, we're reprogramming an enzyme that makes antibiotics. So there are entire BME departments that do this, right? In some places, this is the main focus of those departments. Here, about a tenth of the department does this. Okay, again, our problem is want to put leucine here instead of phi. We want to change 2 out of 9 of these positions. The rest are flexible. And this is about the combinatorics I've already given. 3,000 mutations, nearly 750 million conformations here. And we've reduced this to a scientific programming problem here. So what predictions do we get out of this, right? We just run it. So this is the top prediction to adenylate leucine. There's a threonine to leucine mutation here and an alanine to glycine mutation here drawn like this. This is the predicted GMX structure. So out of 3,000 sequence, this is the top prediction, like it's the best one in the model. That's pretty neat. So that's a prediction, how this would work. Here's another prediction. What if we wanted to change the specificity to arginine? So we ran this for several charged amino acids. This one is a nice one to try experimentally because this one is #1 for arginine and #4 for lysine. So we can make one enzyme and try it for both. Again, many more conformations here because arginine is so big. There's nearly 1 billion conformations here to search through. And you can see these here. You can see the mutations here. There's the D down here. Get rid of this gatekeeper mutation here, G. It's thought that mutating the alanine to glycine kind of opens up the active site, kind of unlocks it in some sense. That's a hypothesis. Okay. What do we do next? So one of the important things in enzyme design is that mutations far outside the active site can be super important. And I don't want to go into that here, but basically it uses iMinDEE plus some entropy measurements. So we can make predictions of mutations that are really far away from the active site that when added to our active site mutations, increase the specificity, binding, or activity. And then we tested these as well. And this uses the same framework, but maybe I'll let you read the paper or take questions about it. It's kind of a cool thing. Second stage. So lots of things you could do here. This is all done in my lab. You have to express and purify the mutant proteins. This is work done by Cheng-Yu Chen. It's now at Arizona State. The computation was done by Evelyn Georgiev, who is now at Vanderbilt as a professor. So you have to make the proteins. You have to do binding assays, activity assays, actually measuring enzyme activity, kcat over km, also stability using CD. And so what do we get? Here's some visualization. So first of all, for each mutant, you get these kinetic curves, right? And you're basically measuring the catalytic activity, kcat. You're dividing this by km, which is basically the velocity at half, concentration at half max velocity. Here we've normalized things, but you can see here we have the wild type here, rather specific for phenylalanine here, kind of interesting. And you have all these different mutants we made. So our best computationally predicted one for the active site is this guy here in yellow. It's pretty specific for leucine, doesn't have much activity for phenylalanine. If we take these and add the distal mutations, so here, we predicted mutations that would strengthen the binding and activity that were fairly far away, tried some of those, added those in, it goes up by, factor of 3-fold or so forth. So the best ones are really quite good. So basically, this is a pure, straightforward computational prediction. If we add to that these sort of stabilizing or bolstering mutations predicted computationally outside like this, we get things that are really quite good. And so this shows that, computational design run fairly purely can go through and predict modifications to the active site that will switch the specificity from phenylalanine to leucine in this case. The activity for charged amino acids is less, but you can clearly see them on these curves. Here's for arginine, mutant versus wild type lysine, can't even measure the activity for lysine. So we can definitely bolster these and get decent activity out of these. So this shows the proof of principle for structure-based computational enzyme design out of a big space. And we only had to try a few things to get this to work. All right. Now, I've already talked about this work on predicting resistance mutations. I believe this was in first or second presentations. And in the interest of time, and also because you've seen it, I don't want to repeat it here, but please go back and look at those papers and that lecture if you're interested. It's also a fairly clean validation of this methodology, but also has interesting biological results combining positive and negative design. But I wanted to tell you about another design story that relates to designing an inhibitor, and how that worked, that has some validation. Some of you read the paper for this as well. So the motivation for this system comes from cystic fibrosis. So it's basically a disease of children and young people. Lots of problems, GI, lungs, and so forth, getting really clogged with mucus. The median age of CF patients upon death is about 35. And it's all caused by mutations in this protein called the cystic fibrosis transmembrane conductance regulator. The structure was just recently solved. So this is a chloride channel. In fact, back in the day, pediatricians used to lick babies' cheeks when they were, after they're being born, to see if it was salty, because you could detect abnormal chloride transport. So this mutation, which is the deletion of phenylalanine codon at position 508, this was discovered a long time ago. Basically, develops a lot of different problems when you have this mutation: misfolding, mistrafficking, and poor function. And interestingly, if you're from the genomics world, this mutation was first reported in conferences in 1987, in papers 1989. It wasn't until 2012 that the first therapies that particularly targeted any mutations in CFTR were brought to clinical trials and to market by Vertex Pharmaceuticals. The initial ones targeted rare peripheral mutations. Only now do they have some that target this actual genetic problem. So this shows that from getting the sequence to getting something translated to therapy can take a really long time, and that required lots and lots of modeling, high throughput screening, hardcore biochemistry and biophysics to take this remarkable genetic finding, which is important because 80% of moderate to severe CF patients have a homozygous mutation of delta F508. So very important, although there are other ones as well. So it takes a long time to develop this into a drug or a therapeutic. Now, I'm going to tell you about designing an inhibitor that modulates some of the trafficking problems. And so this is a PDZ domain that grabs onto the C terminus of the CFTR protein, and it pulls it. This actually pulls it to be degraded. So let's talk about what happens in CF, cystic fibrosis. So because of this mutation, your protein CFTR might not fold very well. And it also might have other problems. So once it gets into the apical membrane, it might have very poor conductance of chloride, although it still works, right? And then it's also rapidly degraded. So these proteins in the membrane are rapidly degraded through constant endocytic recycling. And basically there are these trafficking proteins like CAL, CFTR-associated ligand, and NERF, sodium hydrogen exchanger regulatory factor 1. These have PDZ domain hands, and they grab on to the C terminus of CFTR. NERF drags it back to be reinserted in the apical membrane. CAL drags it for degradation to the lysosome. So these are various things that you could imagine targeting with therapies. So you could have, correctors that correct the folding, some kind of small molecule therapy that corrects the folding down here. You could have stabilizers that try to make the protein more stable so it folds, so it conducts chloride better in the channel, performs better, in some sense. And these would basically be a different class of inhibitor. They would prevent mistrafficking of the protein so that even if you had cystic fibrosis, they would stay more in the apical membrane and continue to work. So it's an orthogonal method of therapeutic intervention. It can be used additively with the other ones. And the other thing to note here is that the mistrafficking of CFTR, i.e. the degradation, is much faster in the mutated protein, in the one that has the delta F508 deletion. And so the idea is, can we design a selective inhibitor that will ablate binding of the PDZ domain hand of CAL to the C-terminus of CFTR, thereby reducing the amount of CFTR that gets taken to the lysosome for degradation. So that's what we'd like to do, competitive inhibitor of the C-terminus. So let's see what we can do with protein design. So again, here's a picture. You know, we now have some structures. There are some structures now, wild type CFTR, but this was a homology model that we made before there were any such structures. So kind of interesting, you could do designs based on homology models. Here, you know, we basically have CFTR here. We have the cal up here like this. The idea is instead of letting cal grab onto the C-terminus of CFTR and drag it away as it's doing here. So here's cal on the C-terminus. It's about to drag it to lysosomal degradation. We'd like to instead design an inhibitor like this. Inhibitor will go into cal, block the active site of cal so it can't find this. That's the structural picture, so to speak. Cartoon picture, but a picture nonetheless. So this is, of course, an interesting exploration of PDZ domain in general. So you could try to design these just based on all the sequence information about these PDZ domains. That would be pretty cool. And we can use that information, but we wanted to try to do this from a biophysics point of view, and then we wanted to benchmark it against sequence-based techniques to show that it could do as well or better. And we actually started with an NMR structure. Okay. So we started the NMR structure of the cal protein binding to the C-terminus of CFTR. Then we took that as information to design a better peptide that would bind more tightly and outcompete the C-terminus. So, you know, the design here has similar kind of setup, right? We'd have 4 mutable residues on the C-terminus of CFTR and a lot of flexible residues, all the things that are nearby, that would be influenced by the mutable residues we have either changing or flexing around. So pretty big design in some sense, a lot of things moving at once. 4 mutable, about 11 flexible residues. And again, we want to mimic and improve on the C-terminus here, starting design like this. So I'll show you some of this data, but we wanted to do lots of validation of this. So one thing we wanted to do is a lot of people are very excited about high throughput screening. Now, high throughput screening for peptides is you design a peptide array. These are arrays like this. Lots of peptides are bound to that. You wash your protein over it, you see what binds. It basically gives you a binary readout in BOUs. But we had several arrays with collaborators where we had between 1,000 and 3,000 peptides on these arrays to screen rapidly for binding. And we wanted to see how our predictions did relative to the arrays. Of course, we designed new peptides and prospectively measured Ki values. Basically, we also measured negative designs, things that were predicted not to bind. And then we tested for biological activity, then later crystal structures, and looking at entropy and landscapes. I'll tell you a little bit about pieces of this. It's all in the papers. So here's a summary of experimental results. Obviously, we have ensembles because it's K-star. So here's what the ensemble looks like for our top peptide, top ranked peptide, which is this sequence here. It's a hexamer. We isolate the terminus, but it's a hexamer. And basically the low energy ensemble looks like this binding. This is predicted by Osprey. Now, here what we've done is we measured binding and we plotted it. We converted the KIs or KDs to delta G of binding. And the thing we're trying to outcompete, of course, is the wild type CFTR sequence. So basically 300 times better binding than the thing we're trying to compete. Now, these domains bind a lot of things, so it also binds tighter, much tighter than the highest affinity natural ligand, which is a 10-mer. And here's all our top predicted peptides. They bind like this, lowest binding with about 2 micromolar binding. All these things bind fairly weakly compared to drugs, but they're still good starting points. So much better than the thing we're trying to outcompete, much better than natural ligand, and they all do pretty well. One does pretty well in particular. This lowest one is shown here. Here's its sequence as a peptide. That's pretty interesting. All right. Now, the other thing we did is we wanted to basically look for a test of the methodology. So we picked a diverse set of the things predicted not to bind, and we tested those as well. And you see these are poorly ranked. Of course, this is a log scale, so these are compressed. These are, all the black things are actually worse on a non-log scale than all the green things, but some are pretty close. You'd be right to think these are sort of false negatives. So this is one way of showing that negative design is actually quite hard. I mean, for a biological or actual physics experiment, this is actually really good. This is very significant that these are all low and these are mostly high, but these are quite close instead. But it shows, the power of the method and also places we can improve things. Also shows how darn hard negative design is. It's really hard to get it right. Okay. Now, here's another thing we did. We wanted, after getting binding, to test this biologically. So we wanted to also compare it to the previous best peptide ever, which was designed by our collaborators here. And his binding is right here, right? So our peptide is about 7-fold better than the best one previously discovered in terms of binding. What about in terms of biological functions? This is tested in Using chamber. There is no animal model for cystic fibrosis, right? No monkeys, no mice, no anything. All you can do is this experiment where you grow up basically cultured human cells. So these are upper airway human epithelial cells, all of which are clones that have a homozygous deletion of phenylalanine 508. And then in the Using chamber, you're measuring chloride fluxes and changes in it relative, of course, to a baseline, which is a CFTR specific inhibitor. So you can actually knock it down to 0 for a negative control. And then you're looking at whether your rescue peptide, which is washed over this, does your rescue peptide enhance chloride flux or not? And this is the classic experiment that's done before moving to actual clinical models, because there's no animal model. You do toxicity and safety first, of course, but this is how you see that it's an ex vivo experiment to see whether or not you are improving lung function in a monolayer of these cells. Okay, so interestingly, our collaborators were super excited about iCal35, but in this experiment of enhancing chloride flux, what happened was the enhancement by iCal35 was actually non-significant, not statistically significant from a control peptide. The control peptide is a scrambled peptide, like a random scrambling of our sequence that we then validated did not bind in vitro. Whereas over here in green, you can see our peptide actually is statistically significant. It is a significant enhancement, about 12 to 15% of chloride flux. And that's comparable, at least at the time, to the best correctors and stabilizers that were in preclinical and clinical trials at the time. And it could, in principle, be used in an additive fashion. Now, what can we learn about methodology? Okay. So in addition to doing this, we wanted to compare to other kinds of algorithms, all available to us. So up here is the sort of cartoon of this. Here are the Osprey K-star sequences, and they're ranked in order, and it's a provable algorithm. So this is the absolute order. There's nothing missing. And then we can run the same exact computation, except using rigid dead-end elimination and without K-star. So this is basically rigid rotamers without ensembles. And this is Osprey K-star sequences with full partition functions and minimized conformations. And what you find here is that you get some sequences, and 2 of our top sequences are actually appear, right? 4 and 6, but the rest don't appear at all. Some other sequences appear. And in fact, if you did this, you would miss our best peptide that actually worked preclinically and miss a lot of other good things as well. Similarly, if you compare the answers for rigid DE and MinDEE, right, you can run these, you can see that in the top 30 sequences, the number of overlaps is very small, about 3 or 4. Overall correlation is very poor here. So most of the experimentally successful sequences were missed by rigid DEE or even MinDEE without K-star. So we have concluded from that and other experiments we've done that you actually need continuous rotamers, i.e. MinDEE, as well as K-star, if you want to design for function, that is, for binding. The debate is still ongoing, but that's where we are on that. I have a few minutes, I'll say a few more things. So one is that these compounds are going ahead in being prepared for clinical trials by a small biotech company, Rhode Island, last I heard. And I don't want to bore you with too much detail, but I wanted to say that one of the things we do as scientists is we try to go further to see why did the design work? What can we learn from it? And how can we do better algorithms in the future? So we have pretty strong evidence here that this is good binding and good biological activity, right? But we want to know, does it serve this purpose for the right reasons? Like, does it bind in the same conformation we predicted here? Let's figure that out by solving the structure. So what we did here is we solved the crystal structure with our collaborators, high resolution crystal structure of the cal protein and KCal01, our top prediction here, and deposited that and published a paper on it. And so you could look at this, it's pretty nice structure, all the details here. And there's a close similarity between the design models that were predicted and the KCal01 and the crystal structure. So we can go back and we can compare the design model. So one of the nice things about the methods you're learning about, they don't just give you a sequence that you can then build, they actually give you a structural ensemble that predicts the mechanism of proposed action, like why should it work? So then we can go back and we can solve the structure and we can compare the design model, which is the reason for why it should work, with the actual crystal structure, which gives us some confidence that it's making the right predictions for the correct reasons. And in fact, we can do lots of comparisons. Here's the ones that we did here. So, there are lots of steps here. We start with the NMR structure. We then wanted to relax away some obvious problems by doing molecular dynamics refinement of this, relax it a bit. That gave us a design input model from this design that went into Osprey. That, of course, Osprey is changing the inhibitor a lot, making a new inhibitor. So we get out an ensemble model, a set of structures that represents the design structure. Now what we can do is we can then solve the new structure of cal with KCal01, our inhibitor. We can compare that to structures of other inhibitors solved by our collaborators and to all these models. And we can come up with comparisons to see how we did. So and you can do quite a lot to look at these details. I'll let you read the paper for that so you can see what the differences are between, again, this is our inhibitor, this is a competing inhibitor, this is a competing inhibitor in a different non-crystallographic symmetry. But one of the things we found was that although you can see differences here and look at them, we really thought the story of why this works better than other things has to do with entropy. So remember, what do I mean better than other things? So here we're out-competing the best inhibitor discovered by high throughput screening of peptide arrays. So the computation is giving you 7-fold improved binding, but also giving you a significant versus non-significant biological change in activity, probably due to that increased binding. We think, oh, that's terrible. Get back. Didn't mean to go back all the way. Okay. We're going to have to do a little search here. I put in lots of extra slides in case there were questions. All right. So to say what I meant to say here, we think the improved binding is the result of this. But looking at the crystal structures, you can kind of rationalize changes in these structures. You can see differences, but it's hard to rationalize exactly what's going on here. We think the story has to do with entropy of binding, and since Osprey models a measure of conformational entropy, we think that's where it's getting its power. And I won't go into details here, but we have a method this is actually in MarkStar. So this is a method of mapping not just what's happening in one energy well, but actually visualizing the entire conformational space. So in other words, when setting up your design, you're saying certain residues will be mutable and certain residues will be flexible. Let me take you at your word and say those are the flexible residues. That will define the possible entropy of your system. In the algorithms I've talked about so far, we've talked about optimizing for both enthalpy and entropy, namely the heat of binding and also the entropic component of looking at those microstates and summing over those partition functions over molecular ensembles and then dividing them to get a quotient representing Ka. And that'll give you, predictions of binding. But the algorithms are actually fast enough that they can actually produce a provably good approximation, not just to one conformation, but to the entire energy space of the design. So in other words, the energy landscape of the entire conformation space. But how do you visualize such a thing, which is very high dimensional? So we came up with this idea. This is Graham and JJ's idea from systems research, ring diagrams in computer science. And we can go into this, but basically it's a way of comparing the bound versus unbound states and looking at the difference in thermodynamics between these different peptides. So roughly speaking, you see a difference between bound and unbound state here. You see the proportion, statistical weight of the GMEC compared to other residues. And what's interesting here is that, you can see that in the bound state, right, the GMEC has a lot of statistical weight, unbound state, not so much. Nice visualization, but we can see differences between the binding of the complex to KK-L01 and versus the competing inhibitors. And one thing about these competing inhibitors, they're also much larger as well. This is a large inhibitor, about 10 residues. So because we can see differences between the bound and unbound states between KK-L01, our inhibitor, and these competing inhibitors, these probably explain why ours has the best binding efficiency. So binding efficiency is basically KD divided by molecular weight. So a bigger thing can bind more tightly, but it's a more expensive drug in some sense. It's paying for it with its mass. So by analyzing these diagrams, we kind of were able to come up with a story about the difference between the entropy of these states, which we think Osprey is capturing. And you can say some words about this, but I'll let you read the paper for that. So basically, we can look at the differences between the internal energy and entropy based on these diagrams. You can also come up with examples from these landscapes that show that rotamers are predicted to be flexible in these states. So, for example, here's a valine that actually populates different rotameric states all with good contacts. And we think that this suggests that in the model, which binds very well, that's actually switching rapidly between these interchanging states. These are probe dots indicating pretty good contacts here. So you can do these analyses of the landscape post hoc based on your crystal structures to try to rationalize what is the entropy that's being used to make better predictions. So in short, this is these are kind of the big points of showing you this, is that, it's using K-star algorithm and validating it, prospective experiments, and at the end of the prospective experiments, some crystal structures to show that we're really getting the structure that we predicted, and that's why the mechanism is working that way. And finally, analyzing the entire energy landscape to explain the binding efficiency. Why is this such an efficient inhibitor? Okay, so I want to stop and be able to have a little discussion if you guys would like to. So in short, thinking of this, yes, for design, we always want algorithms with provable accuracy, right? And here's some examples of those things. We've gone at this point through, K-star and MinDEE a little bit. We might read some of these later or use them. There's some projects involving these later algorithms. So that tells you that what the algorithm is computing are the best answers in the model. So you know about them. But we also want these to be fast and be able to measure our speed. We don't want speed just to be, I ran it and it was fast. We want a prediction of that speed. And there, computational complexity comes in, probably a great intellectual contribution of the 20th century is computational complexity. We can have things that speed up by a constant factor. We can have things that speed up by a combinatorial factor, a polynomial speed up, or an exponential speed up. These could be things that we can measure empirically. We can run it and see that we get these speed ups, or at least show that they're possible. Best of all is the provable speed up, showing that we always get a polynomial speed up or exponential speed up. That's a very high bar. Looking back over here, we can prove that we get these speed ups empirically for a number of these, but to get a provable speed up, we need to go to something really quite fancy like BWM star that looks at sparse residue interaction graphs. The others have empirically the ability to do this, and you can measure it, and you can see why it's happening, but it's hard to tell ahead of time that you get these speed ups. So our ideal is down here, but those are difficult to obtain. And again, to summarize these, we talked about constant factor speed ups. That's the original K-star algorithm, which is in chapter 12. We have a hint of combinatorial speed up. The basic trick here is as follows, and this is the trick that I showed you, is that in the original K-star algorithm, right? And let's actually actually look at this. Go back over here. So in the original K-star algorithm, these energies here are not sorted in any way, right? It's just, you know that they're coming. You haven't computed them yet. And they can be in any order whatsoever. But the beauty of these A-star algorithms is they will enumerate these conformations either in order or in order of lower bounds. So the way to think about this is as follows. Suppose now I have these energies E-min, E1, E2, all the way, all these guys. Suppose these are sorted in order so that EI plus 1 is guaranteed to be worse than EI. And the energies get larger and larger as you go out in the sum. And therefore their statistical weight goes down and down and down as you go out further in the sum. The whole concept here of these approximations is if that's the case, after computing a running sum, this Q star here, I can bound the tail because I know that if I'm at position I and I've just computed E sub I and exponential of minus E sub I, then I know that for EI plus 1 all the way out to EN, all their energies will be worse and therefore their statistical weights will be less. So I can bound the tail. And if I have a way of bounding the tail, I can eventually compare that to epsilon and I can say, you know, the tail doesn't matter. I'm close enough. And then you can just delete the tail without looking at it. That's the key thing. So when I started insisting that it would be very useful to be able to enumerate our solutions in order of energy gap free, it sounded like a computer scientist just wanting to be perfect. In fact, it turned out to enable the computation of partition functions to a provable approximation because the energies come off the heap in order. And that meant that you could bound the tail and get rid of an exponential number of conformations typically that you don't ever need to look at. And this was kind of a remarkable affordance of these algorithms. So it shows that even if you are embracing provable algorithms just because you want to know your answer is reasonable, it can have all sorts of unexpected benefits in the next year or two downstream. And that really is the difference between getting a constant factor speed up and an exponential polynomial speed up here. So that's kind of the sort of key trick there. And if you don't have that, you're just enumerating these things and you don't know what's next. So I think that's how I wanted to leave things for now. And with that having been said, I think I've said enough maybe, and I want to stop sharing the screen. I'll stop the recording and we'll take some questions. Okay, pause recording.